It's a great privilege to have two wonderful speakers today that I'm very much looking forward to hearing from. Um,
Why don't you introduce yourself? Do you mind terribly?
oh, not at all. My name is Ely Iglesias. I'm new to this role. I'm helping Adrian with the Pacific Monthly Call, but I've been a member for JETZON for a while. My work is focused on the effects of oceanographic variability on mesopelagic fish in particular. So I'm really excited to hear about a new aspect of the mesopelagic today. I'm a
I'm
postdoc
a
at
postdoc.
working with Dr. Anela Choi.
Thank you. Um,
Former slug.
Yes, a proud slug.
there we
Um,
go.
And so today I get the chance to do introductions as well. So we have Dr. David Siegel, Distinguished Professor in Geography in the Geography Department at the University of California, Santa Barbara, just down the coast from where I am, who will be presenting on assessing marine snow dynamics during the demise of the North Atlantic spring bloom using in situ particle imagery. And we also have Dr. Sasha Kramer, who I'm so thrilled to cross paths with again, who is now Assistant Professor in the Department of Earth and the Environment at Boston University, who will be speaking to us about predicting carbon export flux from ocean color-based phytoplankton community composition. Did I say that right? Yes.
Good job.
Okay, so with that, I will turn over the floor. Did you guys end up figuring out who's going first?
Oh yeah, I let Dave win virtual rock paper scissors. He's
Okay.
Yeah. Okay,
All
good.
right,
There we
then
go.
Dave, the controls are yours.
Okay, let me see if I can make this work. We didn't test this part.
Sasha and I did a run through, so hopefully the
Oh, okay.
settings are. There we go.
There we go. And where's the little button? There it is. Continue to share using share plays. Stop using continue to share, I guess. Okay, that was a new thing. Okay, I'm going to talk about assessing marine snow dynamics during the demise of the North Atlantic spring bloom. This is work from the 2021 exports cruise to the Porcupine Abyssal Plain. Shown on the left panel are the three ships we used. In the foreground is the Sarmiento D. Gamboa. In the middle is the Discovery, and the far one is the James Cook. So you gonna work? There we go. So this is our the cruise was during the May of 2021. It was near the Porcupine Abyssal Plain. We had a multi-ship, as you saw, and multi-autonomous vehicle deployments assembled to look at all three of the major pathways of the ocean's biological pump. So sinking flux, the migrant flux by zooplankton, there's by zooplankton, and the eddy and the mixing pump. Our sampling focused on a retentive anticyclonic eddy. The idea there was by sampling within an eddy, we would the difference the low potential vorticity in its core would keep the insides of the eddies from interacting with other places. During the cruise we had four intense storms. Um, um, all greater than 40 knots, some with waves in excess of 5 meters. It interrupted ship sampling. It just disturbed the mixed layer and exchanged some surface waters due to Ekman transport. And it's part of the story I'm going to talk about. In terms of how we fit in, how our sampling during May of 2021 fit into the North Atlantic spring bloom, we kind of caught its tail end when we arrived. The initial silicate concentrations were pretty close to zero, although we did have nitrate in the water column. There were lots of diatoms there. So we concluded that we've missed the large part of the export event. And during the cruise, mixed layer, particular organic carbon and chlorophyll concentrations both decreased in time. A lot of this is in a context paper that Leah Johnson has led that was published a couple years ago in Progress in Oceanography. So today's focus is going to be on the assessment of marine snow dynamics using particle imagery. And this is a paper that came out in December of this year. So shown here are vertical profiles of daily mean particle volume spectrum. They're done in differential form. So on the x-axis is size ranging from a tenth of a millimeter to to 10 millimeters, and in a vertical from the surface down to 500 meters depth. There what's and what you can so the colors shown are the intensity of the particle volume, so you get a sense of both abundance by its color and size by where it fits into this. And so if we start and we look from the days go, these are selected days. These are not all the days that we sampled, but it's from May 5th to May 29th in the year 2021. And the little pictures of the waves show when the periods were where we had our storms and breaks in sampling. So the surface particles we're showing things in as particle volumes a spectrum in differential form because it shows more of the nuances rather than showing things as the particle size spectrum. And these are only samples that are taken near the eddy center. So we can see that surface layer particles become more numerous as and the colors emitting and they're growing, so they're moving to the right as a function of time. And this goes contrary to what's happening to the POC concentrations in the mixed layer. Initially, the particle volumes below about 100 meters were low, and eventually about on like the 16th or so we start to see this plume of very large, these are 2 to 5 millimeter sized marine snow particles sinking to the depth at a speed of roughly around 33 meters per day. So what we're going to focus on today is what are the physical and or biological processes that are driving the observed patterns. So let's start in the surface ocean. Shown here is just the clip of the top 120 meters of this. And you can sort of see that in any way you look at it, so the black line here across each of the spectra is showing the depth of the mixed layer. And so what you're seeing then is that the particle sizes and particle volumes seem to increase as we move from the surface ocean down towards the mixed layer depth. And if we look at metrics of particle size, so does this work? Do people see this? I need a yes.
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 Thank you. So what you see is this is calculated from the cumulative probability distribution of the particle volumes, and it's measuring kind of the biggest size of the particles that we see. We can see in the cruise means that the particle sizes go from something bigger than a millimeter down to something more like more greater than 2 millimeters. So there's a doubling in particle sizes as they go from the surface ocean down with this all within the mixed layer. The mixed layer is turning over. Whereas the turbulence levels, which are calculated from scaling relationships from the air-sea momentum and buoyancy fluxes, show a very rapid decrease and almost a two order of magnitude decrease. And so we see an inverse relationship of that. We can look at this on hourly data and show in this sort of space of largest particle on the y-axis here and the dissipation rate here. There's a statistically significant inverse relationship. Also, we can see that this is a relationship that comes from laboratory experiments done by Alice Aldridge and Tommy Dickey and others. And they're looking at what is the size of the largest particle they put aggregates into a tank with different turbulence levels, and then look to see what is the size of that of largest particle. And that ends up being kind of an effective upper bound for the size of the marine snow that we're seeing in the field. So that was a view looking at it just static in time. If we look at this now as as a function of as a function of time, this shows that what's shown here is the wind stress. The gray bars are periods of time which we were not working because of the weather. And if you look at this is the upper ocean averaged kinetic energy dissipation levels in the upper 50 meters. And you can see that this varies from 10 to the minus 5th watts per kilogram to something, by about 3 orders of magnitude. So the question is, what do these storms have? What role do these storms have on the dynamics of the marine snow? And so what's shown here is that same picture in orange or brown, whatever color that is. It is shown showing the dissipation rates, and then we've averaged daily values of the 50-meter mean particle volume. So we take the particle size distributions and we multiply it by the volume of each particle by size and calculate what that total volume is, as shown here. And these particle volumes go, during these, periods of low turbulence, increase quite rapidly. And they grow about twofold in just a few days during these calm conditions. Whereas when a storm comes, it seems that they reset. They go from being a high value back down to a low value, a high value back down to a low value. And so it points to the processes of shear coagulation and shear disaggregation as regulating the marine snow dynamics in the mixed layer. So let's look a little bit deeper into this. And so what we've said here is that the storms seem to reset the particle volume, the particle volumes. We can look at this as a function of size. And so what we see here is initially, so the colors, so this is the average particle volume spectrum over the upper 50, averaged over the upper 50 meters of the water column for each day, starting in May 4th and going out to the 29th. The white areas are places are periods where we don't have samplings because of the weather. And the colors are what that particle volume is. And so with the brighter red that these are, over the greater the abundance there are of particles of that, of that size and of that size. The relationship shown before from Aldridge et al. Is shown as this red line. And so you can sort of see that between the storms, there's an increase in both the amount and size of the largest particles in the flow. When the turbulence comes, it seems to reset itself. These black lines here with the air bars are determinations of the coagulation rate from using the measured size spectrum and a bunch of assumptions, showing that the coagulation rates increase dramatically during each one of these calm periods, and again they seem to reset themselves and start over. If we look at and this is for particles, very large marine snow particles, so bigger than more than 5 millimeters. If we look at the relative, putting this into a model that includes also fragmentation, we can look at the relative controls of these things. That's shown in the blue line here in this lower panel. So periods of time when turbulence is low, and what's plotted here is the fragmentation to coagulation rate for the very largest for these particles, for these marine snow particles larger than 5 millimeters. So you can see when turbulence is low, the coagulation rate is much larger than the fragmentation rate, and the opposite occurs when this has here. So it's showing that storms have this proximate control on the large particle dynamics in the mixed layer. It is the first major point I want to make. So I can't see all the things because of the zoomy stuff. So the question is, so the next set of questions here is well, what causes this plume? We can sort of see that this plume happens and it works its way down. So what brought that to happen? So starting at about the 14th, we can start to see an inkling of this plume starting and then working and then working. Oops, excuse me. Oops, sorry. Working its way downward. And so what we think is happening is that as the storms pass, there seems to be a transport of particles from the surface ocean down below the mixed layer, which is this line here, and it seems to accumulate, and what we're hypothesizing is that is the precursor of the sinking plume of marine snow. So as we can see here. So it seems like there's a kind of a coupled physical particle dynamics pump well, that's going on here. And so what's shown here is, so this is the depth profile in the upper 150 meters of the total particle volumes. And then this is the spectrum, as I've shown before, between, oh God, David, you got to stop that. The spectrum of the water column between 40 and 80 meters. And what you see is again, between the storms, there's an increase in abundance and size of the rain snow. The coagulation rates again are showing these increases. And so what we hypothesize is happening is that as the mix, and it's kind of shown in this diagram here, as the storms come, they'll take the larger marine snow and break them up into smaller ones, but they'll also homogenize the mixed layer in deep. As the storms pass and the mixed layer is shallow, there's a detrainment flux of smaller particles that are still that can make marine snow because they just came from being marine snow, but they're beneath the mixed layer, so they're not being cycled back up to the surface and getting ripped apart. What we think is happening is that as the calm ones grows and hears and we make larger ones that sink fast, and beneath the mixer we produce them. So we produce these large pieces of marine snow as you can see here, and that's what leads to these particles grow because they're sheltered from the high turbulence, and this coupling led to the sinking pulse of marine snow. Okay. Let's talk about what happens now within the deeper in the water column. So what's shown here is the same plot I showed before, but of the within the eddy, and then these are all the cases that we had outside of the eddy. And so the water parcels, and this gray line here is this magic isopyctal that we know kind of caps the top of the well mixed layer with the isopyctal layer within the eddy itself. You can see initially, and we know that the water parcels within this did not exchange with the outside. We've done in the Johnson paper there was a bunch of water mass analysis done to that shows that, and we had a Lagrangian float that was drogued at about 90 meters that seemed to that said it stayed within the eddy the entire time. But the initial particle volumes outside of the eddy were much greater than those inside. And so what we think happens is that there were sinking events that occurred outside the eddy, and because there was not this this low potential vorticity core within the anticyclonic eddy that would keep it from horizontal mixing, these events would mix across. So what we got to see then was this eddy core that was unaffected by previous particle events until our event happened. So it ended up being this kind of this pristine laboratory for us to examine the temporal evolution of the rain snow that followed. So what happened within the mesopelagic is kind of interesting. So in that core, we saw the abundance of small particles. So these are things that are not marine snow, but are still seen by the UVP instrument. These are things between a tenth of a millimeter up to about a half a millimeter, a tenth of a millimeter up to a half. And we can see that, oh, David, you got to stop that. We can see that they increased nearly twofold after May 20th. So as that large particle pulse came through, the numbers of small particles increased. Now we know from a bunch of ways that the small particles sink at something like 10 meters a day. So it's too slow for something to get down to 400 meters at 10 meters a day during the experiment. So, and we also know that the turbulence was likely way too low for the large marine snow to disaggregate. So we hypothesized that biotic processes have to be, disaggregating these large particles into smaller ones. So as our first step in doing this, we wanted to look at the zooplankton data that we had, and we had day-night paired mock nest tows and from that abundance calculations were estimated. And these are just shown what they were for pairs on the 11th, the 17th, and the 26th that we had within the eddy core. and what we can see here is that the zooplankton numbers increased dramatically. This is a linear scale. The other ones, the zooplankton abundance here, I'm sorry, this is the marine snow abundance increased dramatically here. The the zooplankton abundance also increased, but not as much. And what we calculated then was the sinking was the encounter rate between a sinking marine snow particle and the and a zooplankton. And so and we made a very simple theory for doing that, and assuming that the zooplankton can manipulate within a particular foraging zone. And from that, we could see that the interaction rates, which are shown here on the bottom, increased about 25-fold between the 11th and the 26th. So we know that the probability of this occurring increased. Can we quantify it? And so what we did is we developed a very simple model that for two sizes of green snow, something that's green snow and something that's smaller, and we looked to see, and we did this over a range of, we converted the measurements of particle volumes into conserved volumes using fractal theory and a bunch of assumptions of what possible fractal dimensions we might have. The model included temporal changes as well as particle sinking by small and large particles, a transformation of large particles to small particles, and their consumption of both small and large particles. Okay, so we calculated particle means and gradients after converting to conserved volumes and created an ensemble over a range of time and space and depth intervals and assumptions for what was chosen for the fractal dimensions. From that, we had then a whole bunch of we had a system we could solve by least squares, and we could solve for both. Oh, David, both the sinking rate of the small and large particles, but also these interaction terms and these law and the consumption terms. So the results of this are that we can make some statements over this ensemble. We can actually calculate uncertainty bounds too. That the large particles were sinking at something like 70 meters per day. The small ones more like 12. These rates of sinking kind of matched what we could do from feature tracing and work that was done by Elisa Romanelli in her analysis of the marine snow catcher data set that was collected along with this. We calculate we model the loss rates. We get something like 12% per day. It's considerably larger than particle associated respiration rates that was made by Nicola Paul and during the exports cruise, like a factor of 3 times larger, suggesting that something besides microbial losses are driving this. It's probably likely, due to zooplankton. We also see a transformation rate of large particles to small ones. It was always a positive. We didn't in our modeling. We didn't say what the sign of the transformation rate was, but it was always greater than 1. It's consistent with the suggestions of biological disaggregation is breaking up sinking particles and creating small ones. So, in summary, we show here that the marine snow that we saw during the export North Atlantic cruise was highly ephemeral and controlled by both abiotic and biotic processes. Mixed layer turbulence is obviously creating and destroying large particles. These repeated storm forcings enhance the sinking by vertically mixing particles down beneath the mixed layer where they are sheltered and allowed to grow through things by shear coagulation. Biotic process in the mesopelagic create small particles from large ones. And so this illustrates the utility of using, particle in situ imaging from devices like the underwater vision profiler for biological carbon pump studies. One of the important things that I want to stress is that our decision to do to do these experiments in a quasi-Lagrangian experimental design, I think, was critical. Because we know that the temporal changes that we see in particle dynamics is independent from any spatial ones due to isopyctal mixing. There's more to the story that's in the paper. There's more to do. There's changes. We have diagnosed changes in the porosity of main snow. We know things were getting more and more porous as the cruise was going on. We can see changes in sinking speeds that was also modeled. Things that we need to do in the future is we need to quantify this coupled physical particle dynamics pump. I think that's interesting. We need to understand the relationship of what we can see with understanding the particle size distribution, the sinking particle flux to the plankton community and to the other biological pumps. And its links to satellite data, since NASA is our primary sponsor. So the entire data set is available through NASA's CBAS data set. The QR code there will take you to the paper if you're interested in learning more. With that, I will shut up and let Sasha talk, or however you want to do this.
Um, it's up to you guys. We could either take a couple quick questions now and then have more time for questions at the end, or Sasha, if you would like to present and just do questions at the end, either one's fine.
Either way is good with me, Dave. Do you want to do questions now or wait until the end?
Uh, let's wait till the end. How's that?
Okay. Yeah, that's fine with me. Awesome. All right, let's get these slides up. Okay. Cool. Can everybody see the slides okay now?
Looks great.
Awesome. Fantastic. Great. All right. Illy, thank you so much for running this call and having us here to speak today. I think this is going to be a really nice order of operations for especially leaving off on some of those things that Dave just mentioned as more work to be done. I'm going to present on some of those topics that he touched on, and hopefully we can think about ways in the future to bring these two analyses together that were kind of happening in parallel, all with the exports data set. So I'm going to be talking to you today about predicting carbon export flux from ocean color-based phytoplankton community composition. And this is all work that I did as a postdoc at Imagery. It was supported by Simons Foundation postdoctoral fellowship that sadly no longer exists, but was a wonderful source of support. And so I have a lot of other people to thank as part of this work, but wanted to just acknowledge all of my co-authors on this paper at the very top of the talk here. So in the broadest terms, the work I'm going to be talking to you about today can be summarized by saying that new ocean color satellite data lets us observe surface ocean phytoplankton groups from space with higher resolution than we've ever been able to observe on global scales before. But while we have these new surface ocean observing capabilities, we're still not quite sure how surface phytoplankton community composition affects sinking carbon flux. And because some of this work is funded by NASA, but also because these ocean color satellites allow us to observe the global ocean on huge spatial and temporal scales, we're very motivated to know how these surface observations might be able to be used as a predictive indicator for understanding what's happening in the deeper ocean where an ocean color satellite can't see. So to start on this first piece of the talk, the ocean color link with phytoplankton community composition, I like to motivate talking about this by reminding us all of a very fundamental challenge that we have when talking about phytoplankton community composition. We're talking about microscopic organisms. So if you think about the amount of diversity that we have in just a drop of seawater on a taxonomic level with phytoplankton, we're looking at vast different sizes, types, morphologies, but this microscopic level diversity can also be seen from space. So we're trying to find methods that will let us bridge this spatial scale from microns to kilometers and understand how the really highly resolved taxonomic diversity that we see on microscopic scales is represented on these global scales that ocean color satellites make observations, and that we're able to predict these more global scale Earth system processes, including the biological carbon pump. So there are a lot of different ways that we can describe phytoplankton community composition, and most of these different methods were employed as part of the exports cruises, including amplicon sequencing, imaging and flow cytometry, and some of these more traditional microscopy methods that give us a longer history into the observations of phytoplankton communities. And so I always like to say the ideal method for doing this is the one that's going to be most effective for your system and your question. By putting these methods together, you can understand better the strengths and limitations inherent in some of these observations. I'm not going to talk about that in depth today. If it's something you're interested in, I'm always happy to chat more about it. But what I am going to talk about today is a method we can use to try to bridge some of these broad scales of phytoplankton community composition and our spatial observations from space. So in this case, we're talking about high performance liquid chromatography, or HPLC pigments. HPLC is the method that we're using to measure the pigments that are in phytoplankton, which contain multiple different pigments. And some of those pigments can be used for photosynthesis, some are used for photoprotection, and there are specific biomarker pigments that can be used to separate certain phytoplankton groups. Now, if you do any work on chemotaxonomy or using pigments to separate phytoplankton communities, you know that there's a lot of variability in some of the ways that we can link taxonomy to pigments. It's not always a straightforward relationship. But by making some broad assumptions about the phytoplankton groups that we most often find associated with certain pigments, we can think about how those different pigments are able to be used to separate really broad groups of phytoplankton, like what I'm showing you here. What you're looking at now on the right side of the slide is the specific absorption of those different phytoplankton pigments, which absorb light differently across the visible spectrum. And we can use some of those differences in phytoplankton absorption features to separate both between these different pigments, but then also from the pigments to think about differences in phytoplankton taxonomy. And we care about phytoplankton pigments affecting light absorption because this is actually the way that we link from taxonomy to pigments to ocean color. So if different pigments are absorbing at different wavelengths, then we can use that information to tell us about how the color of the ocean might be impacted by different combinations of phytoplankton pigments that are indicative of specific phytoplankton communities. What I'm showing you here is the absorption spectrum. We're not actually measuring absorption from space. We're measuring remote sensing reflectance, which is related to absorption features, including our phytoplankton absorption component, which I'm showing you right here, broken down into pigment absorption. And so if we think about going from an actual ocean color measurement or reflectance measurement to phytoplankton pigments, some work that we've done in the past has demonstrated that we can use hyperspectral remote sensing reflectance data, or what an ocean color satellite is measuring, to model 13 different phytoplankton pigment concentrations. And now I'm showing you a dendrogram, a hierarchical cluster analysis of those modeled pigment concentrations that shows that they match our observations of measured pigment concentrations in the data set we use to develop this model, and that we can reliably separate from both our observations and our models of pigments 5 different phytoplankton groups. And along the x-axis there, you can see those different phytoplankton groups divided up. So when we develop models that are using pigments as the input, we're limited in the number of groups we can detect by the number of groups that can be separated by pigments. But here you can see that we can go from really high resolution ocean color data to a lot more information about the phytoplankton community than we've had before. And we can do this in part because of that hyperspectral resolution that lets us separate these short scale phytoplankton absorption features in the remote sensing reflectance spectrum, which we can only recently do on global scales. So NASA's new Plankton Aerosol Cloud Ocean Ecosystem Sensor, or PACE, launched about 2 years ago now, which feels crazy to say, in February 2024. And for the first time, PACE is giving us these hyperspectral ocean color observations on global scales. So the model that I just showed you was developed all using in-water data prior to the launch of PACE. And we're currently applying that model to new PACE data and validating it with in-water pigment measurements to make sure that the model works as well as we assumed it would from those in-water measurements when we apply it to global ocean satellite data. And while previously we were limited to chlorophyll measurements from space that told us something roughly about the amount of phytoplankton, now with these next generation satellite sensors and new algorithms, we can detect more and different phytoplankton pigments that link to multiple different phytoplankton groups. So, taking that prior work, we have this unprecedented information about surface ocean phytoplankton from space that we can get from PACE, this new amazing NASA satellite. But we have to try to find ways to think about how those surface ocean phytoplankton groups might link to what's going on below the surface, beyond where a satellite can see, and specifically how that relates to the sinking carbon flux that we can observe in situ. So the work I'm going to be talking to you about most in depth today is this piece of it. How do we go from surface ocean phytoplankton groups to sinking carbon flux? And is there anything predictive that we can say from these pigment-based surface ocean phytoplankton groups that we can observe with PACE and use that information to tell us about the amount of carbon that's sinking and how it's getting there? So the broad questions that were guiding this work were, first of all, thinking about the connectivity from the surface to the deep ocean. We wanted to know how different phytoplankton taxa were distributed between the surface ocean and the sinking particles that we were collecting in the mesopelagic. Next, we wanted to think about if we could actually use some of these specific groups to be predictive of the magnitude of carbon export flux. When we think about the models that exist of the biological pump, the phytoplankton community structure component is often condensed down into relatively little taxonomic variability. But with the tools we were using here, we had high amounts of information about that taxonomic variability, so we could look a little more in depth about how it might be predictive or not in this case. And finally, we wanted to look at specific mechanisms of export. So going beyond just a correlation between a specific phytoplankton group and the amount of carbon export, could we say something about how that carbon was getting from the surface ocean into these sinking particles by tracing the phytoplankton from the surface ocean into the deeper ocean? So how did we actually do this? Dave gave a really nice overview of what we did on exports, mostly focusing on our second cruise in the North Atlantic. But there were actually two different field campaigns as part of the exports study. So we had coordinated observations in the North Pacific in 2018 and in the North Atlantic in 2021. Both times we had multiple different ships, a lot of different assets, and many, many different in situ measurements to try to characterize as many of these different stocks and processes that you can see here as possible. So in the piece of the work that I'm going to be talking about today, we're really focusing on how these in-water measurements that we have of the surface ocean phytoplankton community link both back to satellite measurements, things that are detectable from space, as well as these sinking particle observations that we have of things falling into sediment traps. So finding a way to link that observable surface ocean pool that we can see from space with the deeper ocean pool. The data set that I'm specifically going to be talking about today is using phytoplankton community composition detected by amplicon sequencing of a specific region of the 18S rRNA gene. And so since I know a lot of people have different backgrounds on this call, I just wanted to take a moment before I dive into some amplicon sequence data to get us all on the same page about what that actually means. So if we take our hypothetical and completely not to scale phytoplankton cells, and think about extracting the DNA from those phytoplankton cells. We can think about this almost like how you might track DNA through a crime scene in a human community. You're trying to link a DNA sample that you have left behind to a specific individual. And we can do something similar with phytoplankton, where we can take traces of their DNA and use them to say who was where. So in this case, from this specific genome, we can focus in on a gene that has a hypervariable region. In this case, the 18S gene with the V4 region. And by sequencing that V4 region, we get a barcode that tells us something about which phytoplankton were actually present in your sample. So for all of the samples that I'm going to be talking about today, we went through this same pipeline and we sequenced the same region of the same gene to have continuous methods between our different types of samples that then let us compare the types of phytoplankton between these different pools. So our different pools that we had for our sampling of this metabarcoding data, we started with surface water samples to be able to tell us what was in whole seawater samples from the surface ocean. We also had bulk sediment trap samples that were measuring all of the sinking particles at multiple different depths throughout multiple different deployments on each of the exports cruises. And then within those bulk samples, one of the tubes at the different sediment trap depths was also capturing individual sinking particles that were collected in a gel layer. And so I'm showing you here what it looks like to image all of those different particles that are collected over the course of the sediment trap deployment. But we can also zoom in and show you a little bit more what it actually looks like to take an individual particle out of that gel. So for each of the particles that were used during this study, we have information from microscopy about the type of particle. You can see here we're looking at an aggregate. And then when that individual particle is extracted from the gel and we sequence the DNA within the particle, we also have information about the phytoplankton in that individual particle. So we know the type and the composition. So from this study, we had our two different ocean basins that were sampled and all of these three different sample types. And so we ended up with continuous surface sampling over the course of the sediment trap deployments over the course of the whole cruise. We also had multiple different trap deployments across different depths and multiple deployments for each cruise. And then within each of those sediment trap deployments at each depth, there were individual particles that were picked, and using the microscope images were classified into different broad types of particles. So we have information about sinking particles in sediment traps as a bulk metric, but also as individual types transporting different types of material. Okay, so here's our big, very diverse data set. I'm going to try to break down some of the different modes of variability that we had in this data set to answer some of those motivating questions that I started with. So first, let's look at the surface ocean phytoplankton pool. And I decided, even though we had this really high resolution DNA data, that I wanted to try dividing the surface ocean phytoplankton community into pigment-based groups. If we're ultimately motivated to be able to link these measurements back to satellite measurements, basing it in something that can be seen and detected from space seemed important to me. So we're looking here at the broad pigment-based phytoplankton groups of the 18S data in the North Pacific surface ocean data set. And what you can see is that the broad proportion of that data, when we look at the phytoplankton DNA data, was dinoflagellate sequences, although we also had large contributions from some of these other major groups, diatoms and Hecrobia and other silicoflagellates. And while we have big pigment groups represented here, within each of those pigment-based groups from the DNA data, we have really high resolution in terms of our taxonomy. So while these boxes are scaled to the overall broad pigment group, within each big box, the little boxes are scaled relative to the sequence abundance of individual amplicon sequence variants, or ASVs. So you can think of those as all of the different types of phytoplankton within that broad pigment-based group that we detected from DNA. And there's really high diversity in our surface ocean samples in the North Pacific across all of these different groups that we sampled. But because we had specific information about the sinking particles in addition to the surface ocean data, we were then able to directly look at the connectivity from the surface ocean to the sinking particles. So what I'm showing you now is the connection from our surface ocean data to the deep ocean data. If a box is colored in a darker color, that means that it was found in the deep ocean in larger particles. And I just want to point out that Dave's small particles are my large particles. So large is always a relative term. And in this case, we're talking about things that were greater than 300 microns in diameter or 0.3 millimeters to use Dave's scale that he was talking about. So again, if it's colored in a darker color, it was found in the surface ocean and also in a larger particle. If it's colored in but only in a lighter color, it was found in the surface ocean and only in a small particle, so something smaller than 300 microns in diameter and not in the larger particles. And then if the box isn't colored in at all, that means that amplicon sequence variant was found in the surface but not found in the deep ocean. And what we found is that we broadly had good connectivity from the surface to the deep. Almost all of the ASVs that we detected in the surface ocean were also detected in the deeper samples, and most of them were found in this larger particle size fraction. So we assume that those large particles are doing the most efficient export of all of the phytoplankton in the surface ocean, and that DNA is maintained from the surface into the deep ocean. So again, this was the North Pacific surface pool. In the North Atlantic, we had a really different surface community that was dominated by diatoms instead of dinoflagellates, although we still had a lot of dinoflagellates present. But we found the same pattern in terms of the connectivity from the surface to the deep. So we found, again, most things in the surface were also found in the deep ocean, and most of them were found in this large particle fraction. Even though we had very different ecosystems, we had a similar pattern between our surface and deep samples. So from this first piece of the analysis, we know that the most abundant surface ocean phytoplankton taxa were all found in large sinking particles, and that most of the taxa that we measured in the surface ocean were found in a sinking particle and typically in a large particle. So we seem to have this strong connectivity across these two ecosystems from who's living in the surface and who is ending up at depth. But then we wanted to look and see if we could actually use some of that information about who's in the surface to predict how much carbon is ending up in the deep ocean. So in order to do that, we had to bring in this measurement of carbon export, or the POC flux term. And what we did for this part of the analysis was focused on our bulk sediment trap data, where from the same trap depth, we had both a bulk measurement of POC flux and a bulk measurement of phytoplankton community composition. And we performed at first a principal components analysis to divide the major modes of variability in that bulk trap data, again, using the phytoplankton community composition information on the scale of pigment-based phytoplankton groups. And what we found is that we had broad associations between POC flux and two major phytoplankton taxa detected from DNA that can also be linked to pigments. So diatoms and photosynthetic Hecrobia, which includes haptophytes or promnesiophytes and cryptophytes. And then in contrast to this mode of variability, we had a second mode with dinoflagellates and chlorophytes. And one of the first things you can see here is that a huge split is between our two ecosystems. So we had one condition in the North Atlantic in most of our bulk trap samples, and another one that was more dominant in the North Pacific. But when we look at the data this way, we're obscuring a lot more of the information that we have with depth and with time about all of these bulk trap samples. So we decided to look at it and see how the actual flux profiles compared to some of these indicators that were highly correlated with export flux magnitude. So I'm starting out by showing you an example profile from both the North Pacific and the North Atlantic. And the first thing I want you to notice is that we're looking on really different scales. So we had much higher carbon export flux in the North Atlantic, which was what we expected and hoped to find, than we did in the North Pacific. So we have two good end members. And when we add on that phytoplankton community composition information, we looked at a simple ratio of the fraction of diatoms and Hecrobia, which were positively correlated with POC flux, to all other phytoplankton sequences, which were mostly those dinoflagellates and chlorophytes that were negatively correlated with POC flux. And what we found is that for the most part, we had similar patterns in depth and time between this simple ratio of phytoplankton groups and our direct measurements of sinking POC flux. And if you're anything like me, you're looking at this figure and you're noticing that sometimes we actually don't have great correspondence. And actually, we have this really notable deviation in the surface ocean in the North Atlantic, which I think is a really cool opportunity to think about whether this is a deviation from a trend, whether it's telling us something about a measurement error in one metric or another. Or whether it's an area for more exploration and opportunity. So we have this incredible data set, but we also are limited in our space and time observations. So sometimes it works really well, but it doesn't always work perfectly as a predictor. When we look across all of our different deployments, all of the different depths, what we found is a broadly positive linear relationship between this simple phytoplankton metric, the diatoms and Hecrobia relative to all other phytoplankton, and the measured POC flux data. And so we can create just a simple linear relationship and think about why it might be scaled this way and whether this might hold up in other ecosystems. Clearly, in these two ecosystems, the North Pacific and the North Atlantic, we had really different flux conditions, but we also had really different phytoplankton community compositions. And when we looked at each individual ecosystem, we did not see as strong of a relationship. We needed to look across the whole range of flux conditions and community composition conditions to get this really interesting relationship that we see when we put the two basins together. So a big piece of the work that I've been doing since the end of my postdoc, I was just in California about a month ago, extracting a lot more DNA from a lot more samples from different ecosystems because we have these two exports data sets, but we're very curious to see if this relationship holds up in other places and times, or if different phytoplankton taxa might be a better indicator of POC flux in an ecosystem where we have totally different phytoplankton community composition relationships. So from this next piece of the work, putting it together with the POC flux, we find that we have two broad groups that are strongly predictive of carbon flux magnitude, and we can use those to model the contribution of these different groups to our POC flux metric overall. And you'll remember the last piece I talked about was mechanism, our export flux mechanism. And so in order to look at that, we had to look at our individual particle data, which I didn't talk as much about today. So what I'm showing you here are the different particle types in each ocean basin, the North Pacific versus the North Atlantic, that had statistically different phytoplankton community composition when we grouped all of those particles together. So in the North Pacific, the only group of particles that was statistically different in terms of its composition was the salp fecal pellets. While in the North Atlantic, we had three broad groups of particles that were statistically different in their composition. What we notice is that the particle types that were contributing the most to POC flux, which was the salp fecal pellets in the North Pacific, and the aggregates and dense detritus and large, loose, and long fecal pellets in the North Atlantic, were preferentially enriched in the taxa that we found to be predictive for driving POC flux. So we see relatively higher concentrations of diatoms and Hecrobia in the specific particle types that we know were most important for carbon export in these ecosystems. So this final piece of the puzzle, looking at the individual particle data, let us actually put that mechanism piece in and think about how we can go from the information in the surface ocean to a predictor of carbon flux in the deep ocean, but also thinking about how that actually is happening, which can maybe also give us some hints about if this relationship either does or does not hold up in other ecosystems, what might be missing. Is there a different fraction of large to small particles? Do we have different grazers present? Are we ultimately getting a different pathway for the surface ocean phytoplankton to get into the deeper ocean? So since this work was published a year ago, I wanted to just take a moment to mention briefly some of the things that are ongoing with Next Directions. We are continuing to push forward with the relationships between our surface ocean phytoplankton data and the remote sensing data we have from PACE, both with pigments, but also with other higher resolution metrics for community composition. I mentioned that we just extracted DNA from a bunch more samples. We have data from Monterey Bay, from Station Aloha, and from the Northeast Shelf LTER transect, where we're able to look at these similar relationships and methods, but in totally new places to the exports data set. And then ultimately, we're interested in comparing how the predictive relationships that we found here might be useful for either confronting models of the biological pump or updating new models going forward, especially now that we have these new and different predictive capabilities from satellites to look at the surface ocean. So tons of people contributed to making this work possible. They're definitely not all named here, but I wanted to call out a few of them specifically. We also were really lucky to have the support of the whole export science team throughout all of this process, which was incredible, and lots of very generous funding sources as well. So that's all that I had for today, and I'm going to stop sharing so Dave and I can answer some questions for a minute. Thanks so much for listening.
That was awesome. Just anyone online, feel free just to unmute yourself and ask a question.
I'll start. I have so many questions for both of you. I enjoyed both of your talks very much. Today, I'll go back since Sasha's already unmuted. One, I've never heard of Hecrobia before. I had to Google it.
Yeah.
Is
I feel
that
like once you get into the DNA taxonomy world, translating that to names of phytoplankton that you've heard before, I was just so motivated by trying to find things that had common pigments that we could use to differentiate them. So there are non-photosynthetic Hecrobia, but the photosynthetic fraction is Primnesiophytes and Cryptophytes.
okay? The second thing was when you showed the bar graphs in the
Yeah.
last few slides.
Yeah.
I think the way I read your color bars is that a lot of dinos were showing up, but you
Yes.
pulled out the diatoms and the Hecrobia.
Yeah. Great
Tell me
question.
about
So
that.
yes, so with 18S sequence data, dinoflagellates have a lot of copies of the
Oh
18S
yeah.
gene. And so
Okay.
dinoflagellates are often just represented in high proportions in that data set across all samples, which is what we found in this analysis. And we excluded a lot of dinoflagellate sequences from this analysis in lots of consultation with Aaron Jones and Tatiana Reinierson, who were looking specifically at all of these heterotrophic or mixotrophic dinoflagellates or the parasite dinoflagellates. So this was like already just looking at what we assume is the photosynthetic fraction. They were present everywhere in high concentration. And so even though you see them in that relatively high concentration, they weren't informative for predicting POC flux. But having them in that denominator term with the all other phytoplankton seems to be somewhat standardizing relative to the changing fractions of these other phytoplankton groups that we detected.
Hmm.
Really good observation and question, though.
Great. Uh, I'll stop in case someone else wants. And then I have a question for Dave. Keep on going. Norm says
Yeah,
keep on
I
going. Is
think
that what
that's
that
maybe
was?
your question. Yeah,
Okay.
great.
All right, Dave, I'm so glad to see I saw an earlier iteration of the talk, but I didn't catch up with what was going on with your talk. So I'm glad to see the final thing, and hadn't seen your paper, so I'm looking forward to seeing your paper. I didn't catch what your small and large cutoff were.
Um, we said it was things that were marine snow, so bigger than half a millimeter, and things that the UVP saw that were smaller, so it's a tenth of a millimeter, and that's because the UVP only samples, has a pretty narrow range.
Right, but where did you make the cutoff when you did your modeling for the transformation and the
A half.
oh, a half. Okay, great,
Yeah,
great,
yeah.
thanks.
So it's basically marine snow and not marine snow that's imaged.
Okay, great. You may know Daniel Clements did
Sure.
the similar modeling, and we didn't know about that your paper was out. We submitted our paper based on pump particles,
Mhm.
which has a 1 to 51 and a greater than 51. That was our cutoff. And we also modeled aggregation and disaggregation a little bit differently than the way you did it. Now I'm really excited to pull up your paper and compare. Ours is in review right now. We sent it in November, I think,
Okay.
and to compare general trends of what we estimated for
I
aggregation and
mean,
disaggregation.
the thing that was that's super important to remember that I probably should have said out loud is that when you image things that are that big, they're so amazingly porous.
Yes,
And so what you measure on a pump is you're looking at mass.
that's
Well,
right.
yeah, and we're looking at the outside edges of snowflakes. And
And also the pumps, we realized the pumps, especially when the snowflakes got giant, the pumps didn't sample them. They sampled them very poorly. So we like missed that, I think.
yeah, and so so Adrian Bird and I did a bunch of transformations of transforming things from projected volume or projected size to conserved sizes,
Hmm.
playing some magic with assuming what a fractal dimension is, and which has its own horrible sets of assumptions. And
Yep.
we'll go with that. What we decided to do as we did this is say, okay, we don't know what it is really, so we're going to try a whole range of things and create an ensemble and use that to correct to do this. The other thing about the UVP that I need to say out loud is the way that it works is and they your Mark Pishrell and those guys have done a fantastic. It's an amazing instrument because you can get stuff that you couldn't get before in real time. It's just awesome. What the issue is that when they image a part, so they image this kind of small area. It's about a couple centimeters across, about 10 centimeters across. It's about a liter at a time for each image. And they decide what is particle, what is not particle with a set cutoff. Okay. So, and they do that calibration against the standard that they've collected, but they do it all in off of Ville-France. So it's all in the Mediterranean in samples that they have. And I think, and we have some mismatches that showed up between stuff that we could see in the marine snowcatcher and stuff that we didn't see in the marine snowcatcher that I think meant that our particles just became so transparent and so porous. I mean, the porosities were 10 to the minus 6. The solid volumes were just compared to projected volumes were just insane. And from any calculation that we could do against pumps, or any way that we looked at it, it was like that. And so, you know, if the instrument just measured a brightness level, and you could do the things like what Melissa's like Melissa Oman does with her snow cameras, it would be a big step forward. But they were designed 15 years ago, so I'm not going to argue. And it's a wonderful thing. I'll shut up. Other questions?
I got a small one, Sasha. Can you
Yeah.
hear me okay? Okay.
Yeah, yeah.
So did you try when you were looking at this, especially at the very end, the relationship between flux and the taxonomic ordering? What did
Yeah.
it look like when you, for example, separated out the diatoms and the heterobia rather than
Yeah.
lumping them together? Was
Really
that
good question,
if you
though.
just had diatoms, would you get just as good a result?
It's a
Or
great question. And I was just talking about this with Meredith Meyer because she has a really cool data set from the Southern Ocean that we were looking at. And
oh, cool,
in
yeah.
her case, it was the exact same thing that we found. Neither diatoms on their own or Hecrobia on their own were strong predictors of flux. And you needed to have that combination. And when you break down the composition within each of the bulk trap samples, I actually have a really quick figure that might be useful for illustrating that if I can just quickly share it again. Let me see if I can just pull that up. What you see is that it is variable which group is contributing more at different depths.
Hmm.
So what we're looking at now is for each of those bulk trap deployments, you can see the measured POC flux in the black lines and then the modeled in the uh, blue or pink lines, and the actual composition of the six different groups we were looking at, those pigment-based groups for each sample is also shown here for the corresponding trap deployment. And so what we found is that because we had sometimes where it was diatoms contributing more, sometimes where it was acrobia contributing more, we really needed to have this combination of those two
Hmm.
broad groups. And even within that, like those individual trap depths, it was just so highly variable that it was one or the other was not a strong predictor. But it's a really good
Interesting.
question. And we certainly tried a lot of different combinations to make sure that we had the strongest predictor without kind of overfitting the model since that ASV data can be so rich.
Well, that's kind of good news for pace because I mean you're going to get a metric that's going to include all the fucoxanthin variants in the same bands, right? You're not going to be able to separate them out very well, so that's good.
Right. Totally. Right. So, right. Thinking about linking it back to those pigment-based groups, we're going to have a group of red algal pigments basically that we could try to correlate with POC flux. Yeah. Yeah, Dave.
So, just to, sorry,
No, no, don't be sorry.
and I know we talk all the time.
Yeah, all good.
So could you, from a remote sensing point of view, you would want to
Yeah.
be able to look at what's at the surface and
Hmm.
predict what's in the interior.
Yes.
And you
Yeah.
haven't done that yet because
Right.
what you've
So
done
that's
here is just
our,
sample to sample. So,
yes.
so that's your proposal. I'm sorry.
Yeah, no, 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 don't be sorry. It's a perfect question because of that. So what we were able to show here was that we have the connectivity from the surface to the deep, and then those groups in the deep ocean are predictive, right? But our exports phase two grant that we were very fortunate to have funded is actually looking at from the surface ocean, can we create a transfer function from that first
Ah,
surface to the first trap depth to the second trap depth? Where do we lose resolution in that predictive information that we can get from the surface satellite data? And we also, here, I'll just show this also really quickly. This didn't end up in the final paper, but we looked at the relationship. So now what you're seeing is on the x-axis, the phytoplankton group from pigments from measured pigments. On the y-axis, that group from 18S. And
mhm,
then the samples for which we had hyperspectral reflectance measurements from
mhm.
exports ran our model, have the predicted concentration of pigments from the reflectance model, coloring those
I
samples
see.
for which we had a matchup. And so, we see different relationships and there's a lot of offset, but for these three things that we would need to broadly model that, we had relatively good agreement across the two different exports data sets. And the kind of range of conditions we had in the different basins was often really different, but creates this nice relationship over the whole data set. So this was our hope that we would be able to use this as a strong predictor, and that's what we're going to continue to investigate in that phase 2 piece of the project. Yeah, Phoebe.
Sorry to hog. Uh, Dinoflagellates,
Yeah.
so they are not important. And do you think that's okay? One, would you conclude that they're not important? Two, do you think they're not important because they're just so crazy and variable, and therefore they're like screwing any possibility of a relationship?
It's a really good question. I think they're definitely the challenge group across all the different in situ measurements that we have, right? They're kind of always the exception to the rule for everything. So, I mean, I'm sure they are messing some things up, right? And yeah, Norm notes they don't sink, they swim, right? They don't even behave like other phytoplankton a lot of the time. And so thinking about that mechanism piece of it, they're going to add more chaos there. I don't know that I would say at this stage yet that they're not important, but I think they certainly weren't the strongest predictor of flux from DNA data in these ecosystems. And it
They're
would be
present
cool.
though, right?
They're
Because
present.
those
Yeah.
they're present in flux. They're
Yeah.
just not a
Yeah.
predictor for the POC.
Totally, exactly right. So we were finding them in all the traps, we were finding them all different, and so much taxonomic variability from the 18S gene. And the work that Erin has done, which is in prep for publication, looking specifically at the mixotrophs and the parasites, fascinating. And there are some relationships there with flux data as well. So I think they're definitely relevant and important. My question maybe is just like, what type of contribution do they have to be making to an ecosystem to meaningfully serve as a predictor? And we didn't see that here, but maybe we would see it somewhere else. So that's part of what I'm excited to see as we look at other places and times. Yeah.
Great, thanks.
These are both such wonderful talks and very complementary subjects. As mentioned, I study mesopelagic fish, so a very different kind of thing. But I did see that you had salp poop as one of the export pellets, and I'm just curious what your thoughts were on looking at some of those, either predators on particles or some of those animals that might be aggregating phytoplankton on their own and producing their own sinking pellets. I was just curious if you have any plans to incorporate that. I don't know how you would, but
Oh, totally. No, great question. And we do. So Karen Stamiashkin is also a part of our exports phase 2 project, and she's going to be looking specifically at the zooplankton community composition at different depths. And we're going to try to link that to the particle composition that we have from the trap, since we have those individually picked particles, and then think about if there's something more direct there about a zooplankton grazer is present, a certain type of particle is produced. Here are the phytoplankton that were exported through that pathway. So definitely something we're thinking about, but also always super interested to have more links to that level of the food web. So I love that that's something you're thinking about and working on. And if you have ideas, let's talk about it more.
great.
I mean, from the Pacific, we did see that salps had an inordinate contribution to the sinking POC flux. The issue was is that they were so patchy spatially. It's really, you know, I mean, fortunately that they make super intense poops, but that sink fast. But so you don't have a whole lot of spatial stuff that what is going on to so you can link a trap to what's above it
Right.
more easily. But still, I mean, it was very highly patchy, the way that the cell distributions were. And so, with that came everything else. So I don't know how, thinking about how to do that for real. But that particular, and of course, if you ever set up an experiment to do that, you wouldn't find salps, and it would go do something else and mess you over. Sorry.
Yeah, definitely. No, and there's also there's a piece of that with the salps making these larger poop particles that were found pretty intact and fresh in some of the deeper sediment traps, we were wondering if that also had something to do with in the North Pacific specifically, there's a lot of reworking of those particles. And so
Mhm.
maybe there's, they're smaller, they're sinking slower, the DNA is degrading more. But in the cell fecal pellets, which had this strong link to the surface community, they were fresh, they were dense, they were sinking quickly. So again, like all of these things are linked back to the mechanism in a really cool way.
That's awesome. If there's maybe a last question, if anyone has one. Otherwise, I realize we're past the hour, and I'd love to thank you both for wonderful talks. So if anyone has any last questions, now's the time. Otherwise, thank you.
Great questions, everybody. Thanks for listening.
Thank you.
And thanks, Lily, for organizing and managing
Yeah,
Oh, of course.
everything.
Thank you so much for volunteering to give such wonderful talks.
no, she was volunteered.
Yeah.
That's,
Oh, your text said you can say no, Dave.
it did say you could say no. That is true.
Oh, that's so great. Well, those are really lovely talks, and it was so good to hear from both of you. And Sasha,
Yeah.
again, so nice to cross paths.
Oh, yeah, you too. No, I'm really
Yeah.
glad to hear your updates and yeah, talk to you all hopefully soon.
Look forward to it. All right, take care, everyone.
Bye,
Bye.
everyone. Have a
Bye.
good night.